Hello everyone and welcome back to Microbiology. Today we'll be covering Chapter 5, the Eukaryotes of Microbiology. This will encompass three different types of eukaryotic microbes, the fungi, protozoa, and helminths. Fungi are probably most well known for the fruiting bodies that they produce as mushrooms. However, they exist in many other forms, including yeast, which ferment a lot of the food products that we consume. Fungi are really good at eating things that animals can't eat, like the woody parts of plants. We don't really think about fungus as being a major pathogen, but in fact, the prevalence of fungal infections is increasing around the world. This is worrisome because we don't have very many drugs to treat fungal infections, and the drugs that we have are not great. There are actually more classes of drugs dedicated to treating HIV than there are to treating fungal infections. The drugs that we do have are slow acting and usually very hard on the liver. It is important to be able to identify the types of fungus that are causing infections, and one way that we identify fungus is through their physical characteristics. Fungi are chemoheterotrophic organisms that decompose generally dead plant matter. And if you recall, chemo means that they get their energy from a carbon source, from a organic molecule, and hetero means that they get their carbon sources from other organic compounds. We have single-celled yeasts, or single-celled fungi, which are called yeasts. And we also have multicellular fungi, which are called molds and mushrooms. The cell walls of fungi can be made of chitin, and they can reproduce sexually or asexually using spores. There are three types of fungi: the molds, yeast, and dimorphic fungi. So some of the physical characteristics of molds include that they are composed of multicellular filaments called hyphae. We're probably most familiar with multicellular filaments in the bread molds that well, not the mold that can grow on bread. These hyphae may be septate, which means that they are divided into individual cellular units, as can be seen here. And then they can also be what's called cenocytic, where they are not divided by septa, each cell, and it looks more like this. Hyphae grow from the tip, but every part of the hypha can grow if separated. So if you were to break off a piece of this hyphae, the new piece could grow as well as what was remaining. We have two types of hyphae that we need to be concerned with. These are the aerial hyphae and the vegetative hyphae. Vegetative hyphae are dedicated to obtaining nutrients from their medium, whereas aerial hyphae are dedicated to the reproduction of the mold and can form spores, which we'll see how that looks on the next slide. So if you were to look at an image of this petri dish and the medium is here at the bottom, you should intuitively know that these would be where you would find vegetative hyphae, which would be submerged in the medium to obtain the nutrients. And then the aerial hyphae are above, which can have these little balls at the top, which can form spores. In this next slide, we have mycelium, which is a filamentous mass which can be visible to the naked eye, which is formed by an accumulation of hyphae. This particular mass is growing on bread. We are all pretty familiar with this. However, much more elegant forms can be created from these structures. So there is a biotechnology company out there that is actually building furniture from these mats. And so here we have some examples of furniture that was created from the mycelium of mushrooms. Also, Mycoworks produces kind of faux leather that's produced through fungus. So this is another method. Instead of having to grow a whole cow and cull the cow, we can just grow sheets of this mycelium in the lab. And you can create things like this purse right here. Okay, checkpoint 1. Robert Hooke drew this first image of mold ever viewed through a microscope. In this drawing, where would the vegetative hyphae be found? For this answer, you can just write A or B. Checkpoint 2. Unlike the hyphae on the right, the hypha on the left is not divided into cellular units and would be described as blank. Now let's talk about the reproduction of molds. There are 2 ways that molds can reproduce. One is through fragmentation, which is like when a piece of a cactus, as a pad of a prickly pear, for example, breaks off, but then takes root and starts growing a whole new cactus. So we can imagine in the process of fragmentation, the very tip of this hyphae could break apart and start its own multicellular organism. This is always asexual. There's no genetic recombination of gametes or anything like that. This is just a clone from this specific cell. Next, we can have sporulation, and this is where spores form an aerial hyphae. And this can be either asexual or sexual. Here is an image of that aerial hyphae under the microscope, and we have the formation of spores within it. In this slide here, you'll see the genetic cycle of sexual sporulation of molds. If we could start over at the spores section on the right-hand side, we'll see that these spores can germinate, and a multicellular mycelium is formed from this, as seen here. The mycelium is actually haploid, and if you need a review of biology, mitosis, and meiosis, I will post some slides under this unit for a little review if you would like to do so. But if you remember what haploid means, then you should sort of understand that process. And so mycelium cells are only haploid, which is different than, say, our own somatic cells, which are diploid. And what happens is plasmogamy occurs, where 2 different haploid cells fuse together to form one cell. And this cell becomes heterokaryotic, which means that it has 2 or more nuclei. Remember that eukaryotic means true nucleus, pro means pre-nucleus, and hetero means like different. So these are 2, in this image right here, we have 2 different nucleuses. Both of them are haploid. And this heterokaryotic stage is followed by karyogamy, where the nuclei fuse together to form a diploid or 2N zygote. And here's the zygote right here, and we see it has now a single nucleus. And from this step here, meiosis takes place, which then forms our haploid 1N spores, and this process repeats again. So in this slide, we need to keep track of a few things. We need to keep track of the names of the different intermediates. We have spores, mycelium, heterokaryotic stage, and then zygote. Spores are haploid. so are the mycelium, and so are the heterokaryotic stages of molds. The point in which it becomes diploid is only the zygote. Now here we have asexual sporulation of molds. Remember I said it could be asexual or sexual. And this skips the process of going from haploid to diploid and meiosis occurring. Rather, we just have spores that germinate into mycelium, and mycelium go through the process of mitosis to make more spores. Asexual spores have a greater clinical significance than sexual spores because they are the only kinds of spores we observe. All right, let's talk about physical characteristics of yeasts. Yeasts are single-celled and they are non-filamented. In other words, they have no hyphae. Here's an example of how yeast reproduction occurs. Most yeast reproduce through a process called budding. And bud is initiated on the parent cell. What occurs is the parent cell's nucleus divides, and then one nucleus migrates into the growing bud. As you can see in this image here, the growing bud, if we look at these two asymmetric spheres here, the larger one was the parent, and the smaller one, the bud that's forming, is going to be the daughter cell. Now, cell wall material is synthesized in order to divide the bud from the parent. And eventually the bud breaks away from the parent. And we can see the remnants of this budding process by looking for bud scars, which occur on the outside of the cell. So each one of these marks is where a septum was formed and a separation of a daughter cell from the parent cell is completed. So now we have the dimorphic fungi. These fungi can grow either as a yeast or a mold. As seen in this image here for Mucor indicus, there are yeast-like growths with single cells and mold-like growth with hyphae that form. The form of which depends upon environmental conditions, such as temperature, CO2 concentration, etc. For example, Mucor indicus, when grown on petri dishes, tend to have a mold-like growth on the agar plate, and this is induced due to high concentrations of CO2. Remember that yeast are they do a lot of aerobic respiration, preferably, and so if you have a collection of them fixed on a solid medium, then the amount of CO2 is going to go up as respiration occurs. And the mold-like growth ceases at the surface where you have more CO2 present, and you'll see more yeast-like growth occurring at the surface. Dimorphic fungi is really important because it's a lot of pathogenic fungi exhibit this trait. Okay, so we've talked about fungi, now let's talk about protozoa. Okay, protozoa are single-celled. They are extremely diverse. They are heterotrophic, so they're getting their food from other organic molecules, and they consume particles of food from the environment. Most of them are non-pathogenic. However, the pathogenic species can be parasites to humans and other hosts. Before we go further, let's talk about symbiotic associations. Symbiosis just means that it's a process where two organisms live in extremely close association with each other. We have three types to discuss. One is mutualism. Mutualism is what it sounds like. It's when a interaction is beneficial to both individuals. An example of this is E. coli living in human intestines. The human intestines provide shelter, food, a stable environment, and in exchange, E. coli can provide important vitamins like vitamin K. Next, we have commensalism. Commensalism is just one of the individuals benefiting, and the other is not affected. An example of this would be Demodex mites that live in human hair follicles. Of course, the Demodex mites are benefited by living off of the cellular debris and having a habitat to live in, but humans aren't really affected by their presence or lack thereof. In general, there are special instances where people might have reactions to Demodex mites. Next, we have parasitism. Parasitism is where one individual, the parasite, benefits, and the other, the host, is harmed. An example of this is Giardia intestinalis, which in human intestines causes diarrheal disease, Giardia. Okay, with parasitism, parasites they tend to have a very complex life cycle, which typically has more than one host. There are two different types of host categories. One is a definitive host category, and this is where the parasite reaches maturity and can reproduce sexually. The intermediate host portion is where a parasite infects in a developmental form and it's incapable of sexual reproduction. Some species can produce a protective capsule that is called a cyst. So here we have a protozoan encystment in Entamoeba histolytica. In the cyst stage, they are resistant to poor food, moisture, oxygen, and temperature conditions. And many parasitic protozoans are transmitted in this cystic stage. We have a select number of protozoans to discuss. These are the amoeba, paramecium, giardia, and trypanosoma. Okay, here we can see amoeba crawling around. Amoeba move through pseudopods. Pseudopods are what we call false feet. They are able to do this through a process of using intracellular filaments that stretch and pull their cytoplasmic materials into a new location. So if it was possible to zoom in here, you would see these filaments pulling up and into this material and kind of coming back like a fountain. But that's not really super important for this lecture, and you're not going to be tested on that, but you do need to be aware that amoeba travel through the use of these pseudopods. Some amoebas are parasitic, like E. histolytica, which can cause an infection of the small intestine and causes amoebic dysentery. Another one is Balamuthia infection of the brain, which can cause amoebic encephalitis. Some amoeba are what we call cellular slime molds. Cellular slime molds are amoebas that can aggregate together when conditions are unfavorable. Slime molds are a perfect example of what a mess DNA evidence has made of traditional taxonomy. Slime molds were once thought to be fungi. Their slime name comes from a part of their life cycle where they can look slimy. However, they are actually not fungi at all and not even closely related to them. They also can have what appears to be a fruiting body or like a mushroom-like appearance, but again, they are not related to fungi. Okay, so now I would like to share with you a video on how these slime molds actually look. So here is a view with the naked eye, so to speak, of an aggregation of these slime molds. And these are single-celled organisms that come together under conditions where there might be low nutrients. So they do this through a process of quorum sensing, where they might sense other slime molds around them, and under certain environmental stimuli, they come together and they form this temporary super organism. Really cool. And what they'll do is they will actually go around and kind of like on the prowl hunt for food sources. Also, something that's very interesting with these is they can form a base, a stalk, and a head and explode at the top in order to release some of these cells to other environments. And in the process, some of the cells might sacrifice their lives. And this is a very interesting process because these slime molds are single-celled, so it's almost as if they're not looking after their own interest, but the interest of the community. Okay, we'll stop there and go back to the PowerPoint presentation. Now next we have the paramecium. The paramecium are called unicellular ciliates, and they have short extracellular appendages called cilia, which are used for propulsion and feeding. None of the paramecium are known to cause human disease, and they can reproduce through a process called conjugation, where two cells attach to each other and haploid nuclei from the two cells merge and multiply to give each cell a new genome. Okay, now we have Giardia. This is the most common human intestinal protozoan parasite in the US. It has, in this image, you can see a sucker disc which allows for attachment to the wall of an intestine. And it's secreted in cystic stage through the feces. And this image right here is of Giardia intestinalis, which causes Giardia in the US. Next we have Trypanosoma. So here are the Trypanosoma. These are blood parasites that are transmitted by insect bites. They typically have long bodies propelled by undulating membranes, and so you can see in this image of Trypanosoma brucei, the blue images are the Trypanosoma. We have T. brucei, which causes African sleeping sickness, and they are transmitted by the tsetse fly. And then we have the T. krusei, which causes Chagas disease. This is found in Central and South America, and it's mostly spread by kissing bugs, which can lead to swollen lymph nodes. But this can eventually lead to heart failure in some. And lo and behold, kissing bug is confirmed to be in Arizona with a CDC warning here, although I don't know of any cases that have actually been reported. This brings us to checkpoint 3. A free-living protozoan is isolated from a pond and observed moving by the extension of pseudopods. What group does it most likely belong to? Now that finishes our discussion of fungi and protozoa. And lastly, we have the helminths. This is a real image of a tapeworm's scolex that was a finalist in the 2017 Nikon Small World Image Competition for microscopy images. Pretty terrifying, right? So helminths are multicellular animals. They have eggs and larvae and are microscopic, but the adult's forms may not be. They have many parasitic species that have complex life cycles. and they can be either dioecious or monoecious. Those of you who have studied plants are already probably familiar with these two terms. Dioecious means two houses. So this means that the male and female reproductive parts are in two different organisms. Monoecious means one house. So the male and female reproductive parts are in the same organism. We have different types of helminths. In helminths, we have the flatworms and we have the roundworms. With the flatworms, we have two subtypes we need to be concerned about. These are the flukes and tapeworms. So let's begin with flukes. They have leaf-shaped bodies, and they have ventral and oral suckers that hold the worm in place. Seen in this Chloronychus sinensis, which is also known as the good old Chinese liver fluke. They lack a complete digestive system, and so nutrients are absorbed from the environment. Flukes have a highly complex life cycle. Here's an example of one. I am not going to try and pronounce the name of this fluke, and it's not necessary to memorize this life cycle, but it just goes to show you that there's many steps involved. The adult fluke releases eggs into the bronchioles of a human lung, and the sputum contains eggs, and these eggs can be swallowed and secreted in feces, and if the eggs find their way into the waterway, they will hatch and form into free-swimming ciliates, and then these ciliates can infect a host, which is a snail, and then they can be also consumed by crayfish, which are predatory for a lot of snails, and in the crayfish, they can become encysted, and the cysts get into humans if the crayfish is eaten raw by the humans. So this just goes to show you just how complex these life cycles can be. Next, we have the flatworms. This particular one is the tapeworm, which has suckers and sometimes hooks on scolex, or the head, in order to hold the worm in place. They range from just 1 millimeter all the way up to 10 meters in length. Remember, a meter is about 3 feet. They lack a digestive system, so again, they absorb nutrients from the surroundings. And the body is divided into segments that are called proglottids. They are produced as long as the scolex is alive. So if this tapeworm was to break at some point in its body, as long as it has a scolex, it will continue to survive. Okay, checkpoint 4. Is C. sinensis dioecious or monoecious based on this image? Now we have the roundworms, and the roundworms range from 1 millimeter up to 1 meter in length. They are the most abundant animal group on Earth, having 4 out of 5 animals as a nematode, which is a roundworm. They live in virtually all environments, and only some are parasitic. N.A. Cobb had a very interesting description that I have here posted on the right if you would like to read about just how common these organisms are. They are different from what we've covered so far in that they have complete digestive systems, and they actually are the most common cause of chronic infectious disease in humans. We have the Ascaris lumbricoides, which infects greater than 1 billion people. Pinworms, which infects 11.4% of US, and including 30% of children in the United States. Then we have the whipworm, which infects greater than 1 billion globally. and the threadworm, which infects 30 to 100 million people globally, and next the hookworm, which infects more than 500,000 people, or sorry, more than 500 million people. So here are some select roundworms. We have the guinea worm, which is transmitted by drinking water that contains water fleas infected with guinea worm larvae. We have no symptoms from this until eventually the worm emerges from a blister under the skin over the course of several weeks. We have the Bancrofti, which are transmitted by mosquito bites and clog the lymph nodes and can result in elephantitis. So as you can see here, this is an example of elephantitis. And then we have the Loa Loa, which is transmitted through fly bites, and the worm develops in the eye. And finally, we have checkpoint 5, a parasitic worm with an incomplete digestive system and oral suckers, is isolated from canine feces. Is this worm likely a roundworm or flatworm?